All right, good morning, everyone. Welcome to GE Current, powered by Daintree. This is our second webinar on horticulture lighting. We're excited to talk to you about what's new and developments that are going on in the horticulture industry. One of the discussion topic that we're going to have today is really talking about the importance of finding the right light. And so the idea here is the science behind the light, but really more importantly is understanding what is the idea of quality of light versus quantity of light. These are two concepts that we would like to just further dive into, because these are questions that we always start talking about, is that a light is not just a light, and try to understand how one light may differ in terms of its quality, but also from its quantity in terms of how many micromoles we're delivering to a plant. And so before we get started, we can do some brief introductions. So my name, Bruin D'Amico. I'm the product manager for horticulture lighting at GE Current, powered by Daintree. And so my role here is really to help work with our customers and try to guide them through our catalog of products and trying to work with customers to get the right solution that works well with their growing either indoor or greenhouse or whatever lighting needs they need. Today with us, we're really excited to introduce Hans. He's our plant scientist. Hans, if you could just give a brief introduction.
Sure. I just came on board. I have a master's from the University of Arizona, where I studied at the Controlled Environment Ag Center, focusing on vegetable grafting and seedlings. That's where I got my first experience with greenhouse technologies. But more recently, I have a PhD from NC State, where I focused on light physiology and how light specifically impacts the plant in a vertical farm setting.
Great. And we're excited to have Hans on board. I think he brings our expertise to a new level, and excited to work with him, and excited to work with customers and also expose our customers to his knowledge. We also have with us Eden Zivik. Eden Zivik is I would say a veteran of our GE team. So Eden, please, if you could just introduce yourself to the audience.
Hello, I'm Eden Zivik. I'm the technology architect working for Current, GE, and all the other company that was before. I've been with the business for 21 years and developing lighting product for using LED. So I work in almost all the application, and now it's good that we have working on horticulture. It's a new challenge. It's really fun. So Bruno, let's start.
Yep. So just for today, we'll just give over a brief agenda. So where we want to focus on our two higher level agendas is really focusing on the plant itself and then understanding how light interacts with the plant, or how the plant receives that light. So we'll be talking about again, quality versus quantity. We'll be talking about how a plant uses light in terms of information, and we'll get a little bit more into there. And we'll talk about how it receives that information through its photoreceptors. We'll also be talking about fixture design. So it's important to understand what a plant needs from a lighting perspective, or from a micromole perspective, from a spectrum perspective, but how you actually deliver those photons to the plant really dives into how that fixture is designed and how you're going to light based on those given applications. So we'll dive into the application considerations, and also a notion of understanding how spectrum versus efficiency and how those are somehow related, and understanding a little bit of those trade-offs when we're walking through. So I guess where we want to get started is maybe, Hans, you could walk us through when we're talking about quality versus quantity, what that actually means.
Sure. They are two very different things, but both convey information to the plant. So when you're talking about quality, light is radiation. And that radiation that is visible to us, we describe it as colors. Now, those different colors, they can provide information to the plant on its environment, on its surroundings. So the plant then uses different light profiles, or those different qualities of light, to interpret the environment and then respond appropriately. And then if you're talking about light quantity, that is specifically the photon intensity intercepted by the plant on an area basis in a given time. So basically you're thinking in terms of a rain collector. If you're going out there with a rain gauge, how many raindrops are falling in to that rain gauge, and then you can measure and quantify how much rain is hitting that certain area. Now you can do the same with light, but instead of raindrops, you're talking photons.
Okay. And so, like, looking at in terms of quality, so we're looking at the quality of light being, what's the range that typically we would talk about in terms of the quality, in terms of wavelengths, I guess?
Sure. So right off the bat, you talk about visible light. And you can break that down into 3 different colors, generally speaking.
Mm-hmm.
And that's your blue, that goes from 400 to 500 nanometers. Green, that goes from 501 to 600 nanometers. And then red, that goes to 600 to 700. And then that's considered photosynthetically active radiation.
Okay.
Even though it goes a little bit beyond those ranges, that's pretty much the giant in the room when we're talking about photosynthetic active radiation.
Okay. And so, is there a reason why, where does those bandwidths, right? Why are we breaking it down, I guess, into red, green, blue light?
Yeah.
So where does that come from, and why are we so focused on, I guess, those 3 bandwidths? There's a little bit that spills out, but where does that come from?
So where does that come from?
Yeah.
That comes, well, first off, we're humans, so those definitions are assigned by the colors
Right.
of what we see.
Right.
And then in a very generalized form, those colors have different impacts on the plant.
Okay.
So a light fixture with high red content is theoretically assumed to drive photosynthesis at a much greater rate due to the classic McCree curve.
Okay.
That's not always the case when you're bringing in all these other variables and factors, but that's one target, for example. But then you also need the other colors or multiple colors to convey information, as I mentioned earlier, to the plant, so you can either restore normal plant function or just maintain normal plant function.
Okay. And so if we're talking about a plant receiving information, so how does that actually work? So we're talking about, let's talk about outdoors. Let's call it nature, right? Let's not get too close to our LED fixtures right now, but in nature, the sun radiates light.
Sure.
That light is, going back to what we're talking about here, this is radiated energies. And so now this light travels, let's say, from the sun, now it hits the plant. So what's going on in the plant? How is that plant actually receiving that information?
I get excited talking about this stuff because plants are almost like supercomputers that we know very little about.
Okay.
So, when the light is first hitting the plant, let's talk about the start of the day. And blue light is very crucial. That can help the plant realize that it's the start of the day. And it will actually, once the plant perceives blue light, it will open up its stomates. Not necessarily knowing that photosynthesis is happening within the plant at that point, but knowing that it will quickly start. So those stomates, they allow the exchange of CO2 and water, and by allowing CO2 to basically prime the pump of photosynthesis, they can start photosynthesizing as soon as the right amount of light intensity hits the plant. So there's step one, that the plant knows that the day is starting, and that's some information given by blue light. Okay.
And so within the plant, structure, right, where is this, it's not just a leaf, right? There's more to a leaf or to a plant. So where does that light actually go? Those are going into photoreceptors.
Correct.
And then, so talk to us more about what is a photoreceptor? We talked about this earlier.
Okay.
What is a photoreceptor, and how does a plant use those photoreceptors to, take in that information and translate that into an action?
So yeah, plants are constantly using photoreceptors to adjust their model of growth throughout their life cycle and even throughout the day. And photoreceptors can be present in the seed, in young leaves, in young plants to plants that are basically at the end of their life cycle. And a photoreceptor, backing up one step here, is basically a protein or a chemical compound within the plant that can perceive a certain wavelength of light. And that's where that quality aspect comes back that we started out on. And by perceiving those different colors of blue, green, and red, even far red and UVA and UVB as well, photoreceptors can help steer the plant in a manner that can make it more successful or more fit.
Okay. And so when we're talking about photoreceptors and we're talking plants, we're talking in generalities right now, but so how are photoreceptors differ from cultivars to cultivars, or are they kind of all the same?
So typically photoreceptors are found in every plant. There are extreme mutants where some classes of photoreceptors are not present. So that's useful in the lab for research and learning more about the functions or loss of functions of these photoreceptors, but not necessarily in a greenhouse or farm operation that wants to make sure that the plant has as many tools as possible to respond to the environment to grow as best as possible.
Okay. And so a plant is taking its photoreceptors, so I guess what is in general, like how many photoreceptors are we talking about?
Sure, yeah. Sure. So depending on the scientist you ask, there are as many as 5 to 7 photoreceptors, possibly even more. And there are 2 major ones, and those are basically like the cryptochromes, the phytochromes, and phototropins. With those 3 photoreceptors, you can gain most of your normality or your plant functions just by balancing and controlling those 3 photoreceptors. Then there are others like UVR8, which is the more recently discovered photoreceptor, and Zeitlupe, which is another one that helps control circadian rhythm in the plant, or telling the plant how long the day is, when to shut down for the day, and then other longer term functions like when to flower and when not to flower.
Okay, so when we're talking about photoreceptors and we're talking about the cryptochromes and phytochromes, I guess there's like in terms of like when you're seeing a plant, right? Just seeing like I guess the way we interpret is a plant, healthy. It's growing. It's either flowering or it looks like the leaves are in a healthy are healthy. The plant has. So some cryptochromes or photoreceptors are taking information and giving growers the visuals that I'm healthy, and then there's other photoreceptors that are giving other things that are less tangible, I guess, in terms of what the grower is seeing in the plant. So can you walk us through let's go a little bit deeper, I guess, into photoreceptors and try to understand from a grower standpoint what they can see tangibly and what they can't see.
Well, let's start with, so basically these photoreceptors are the eyes and ears for the plants in terms of light. So for the cryptochrome, that sees UVA and blue, and that perceives the ratio of blue and the amount of blue and tells the plant, okay, how to respond in those certain scenarios. Then you have the phytochrome, which spans a very large range from blue, red, but has a high sensitivity to red and far red. And the phytochrome is a very crucial photoreceptor for controlling, for example, circadian rhythms, or more commonly known as like flowering period and flower timing. So, they're very crucial. And maybe we're not actually seeing the effects from a grower's point of view, but the plants are able to use those colors to determine their functions. And
so I guess when we're talking, again, thinking about a photoreceptor as being, I guess, like a switch, right, that activates some sort of response to the plant?
Yeah. So you'll have, getting down to the molecular science of it here, but basically these photoreceptors will hit switches on and off to control stem elongation, leaf expansion, even source sink relationships, so where to send carbohydrates within the plant and stuff like that.
And are all these photoreceptors active equally throughout the entire life of a plant, or early on stage development, there's certain photoreceptors that are more sensitive versus the end, or when the plant is getting to a more mature state?
That's a really good question. And the answer is tough to generalize for all the plants. But in terms of photoreceptors, the phytochrome is always present in the plant. And very interestingly, it's even included in the seed. So we don't perceive seeds as something that sees light or doesn't see light, because we just think of it as something with a hard seed coat around it. But it still has a phytochrome in it that can perceive the red and the far red ratio, for example. Now, other photoreceptors are not necessarily innate in the seed, so they develop as the plant gets older.
Okay. And so I guess if you think about a photoreceptor activating a response in a plant, what are your thoughts? Like if we go now from a photoreceptor thinking about the plant going into now the spectrum of light that that's receiving. So in nature, you have the sun that kind of gives a broad, like a spectrum of light that spans, a large span of visible and non-visible wavelengths. But now once we, bringing this along and thinking about how we provide supplemental lighting in certain cases or sole source lighting, how would you take on the subject of tailoring a spectrum of light to photoreceptors and how to get the response from a plant in a controlled environment? So how do you play that mix?
Sure. So there's, looking at the big picture of things, plants have been evolving hundreds of millions of years. And now it's up to us. The challenge is to try to use the photoreceptors present and use them to our advantage. So you mentioned sunlight. And, there's a deliberate or determined ratio blue, green, red, far red. Now, when plants are growing next to each other, they cast shade on one another. Now, that will change that ratio blue, green, red, and far red as compared to what it would be in an open sky setting. And what is happening at that point is you're basically getting an increase in greens and reds in the lower parts of the canopy because the upper part of the canopy is absorbing the blues and reds. So as you're going down through the canopy, that light profile changes, and that is now telling the plant, okay, we're being shaded now, we're competing, we're in a race here, we better change our strategy and be the plant on top. So just by changing the ratio of example, that green and far red, you're telling the plant to, okay, maybe elongate your stem a little bit more to make sure you're taller than your neighbor, and maybe, try to become more robust and expand your leaves so that you can capture more light in those lower light zones and also maybe outshade your neighbor. Now, that sounds like an arms race in nature, but bringing it back to the field conditions, that could be used to the grower's advantage.
So I guess what you're trying to say here is plants are using their photoreceptors. Some photoreceptors are going to take that plant information and it's going to say, I'm sensing a certain color of light, I'm being shaded, I need to change my response.
Exactly.
But once we get into a controlled environment situation, we could use spectrums to force that behavior to our advantage.
Exactly. Yeah.
Okay.
So by mimicking certain light profiles, we can trick the plant into assuming that it's in a low light setting, but it's actually being given just enough light to grow normally.
And so I think that's where it's important. I guess, the whole point of this webinar is just really talk about quality versus quantity. I think that's where you kind of bridge the two, is just understanding from a plant physiology what's that information, the spectrum of light that we need to give to the plant in order to elicit a behavior. So those behaviors are being, taken in by photoreceptors and are being activated. But it's really important to not actually create those by a lack of quantity of light. And so then, if we're thinking about quantity of light, when we're thinking about, fixture design, but really specifically the application, and we start from the plant, what it means, can you walk us through in terms of application how that challenge is approached, let's say in a greenhouse application, how we would merge the idea of spectrum, quality of light, and how we would make sure that we are addressing the quantity of light and how we approach that challenge in a greenhouse?
So yes, in the greenhouse, it's more close to the outside nature. So in the nature, plant will grow normally. Now, the reason why we develop greenhouse is to extend the season mostly, or protect our crop from the outside environment. Sunlight has full spectrum, but let's say if you look at the sun when it's at zenith or if at the horizon, spectrum is changing. There's when the sun comes down at the horizon, there's more red into it. When the day are shorter on the more close to the pole, then you change the spectrum. So even if you're outside or you're in a greenhouse, your plant structure will be different in summer versus winter. So this is where, and also the other thing is the day length that we may not have talked too much, but the photoperiod may affect also the plant growth. So when we bring lighting into greenhouse, this is when we start controlling or affecting the quality of our crop with light. So let's say just because if you're growing in north of Quebec in the winter, there's a lot of red. Photoperiod, the day is really short. Intensity is low. So your plant is kind of competing, like Hans is saying. The plant perceives this signal as I'm too small. I need to grow. I need to go above the other and be the strongest plant. So the structure of your plant will be more elongated or stretched. When we bring light in, then we can control that physiognomy of the plant and have it, let's say, behave or grow more normally, like if it's in the summer. So this is what greenhouse lighting is doing, is we work with the current environment, so sun is there, but we complement or we supplement to the sun lighting to get a more standard or normal growth.
Okay, and so let's say we're talking about a greenhouse, large structure, and we go off like what Hans was mentioning about photoreceptors either interpreting that information as being shade or not shade. What are the challenges in a greenhouse that we need to overcome in terms of a fixture design that allows us to make sure we have either a uniform light level or a high enough light level, but also not impeding, let's say, on the structure or physically creating shade in a greenhouse. So what are some challenges from a fixture design that we need to take into consideration?
So again, from a fixture design, it's strange to say, but the biggest, competitor to our fixture is the sun. The sun is free, sun is there. So we need to make sure that in a greenhouse design or infrastructure, we don't block too much of the sun. So this came into the size of the fixture and also maybe the weight. If your fixture is way too heavy, then your structure needs to be bulkier and may hide more sunlight to the crop. So this is the two main, challenge. The other challenge is, if you're creating let's say a 10,000 watt fixture, even if it's HPS or LED, then it's less fixture in your greenhouse, yes, but the distance between the luminaire will be longer. So that means uniformity will be more difficult to achieve on the crop. So some plant may see higher intensity or the light coming from high angle, where some other plant in the same growing surface may see higher intensity and a light coming from straight up. So yeah, there's some challenge on the design of the luminaire for greenhouse, ideally being compact and bring the light uniformly across or over the surface.
So I guess when we're thinking about a greenhouse, there is a certain level of dynamic light management that needs to be taken into consideration. That dynamic portion being the sun, because you're not entirely you're controlled to a certain extent, but there's only so much control on the sun that we could implement. So now that if we're thinking about greenhouse and dynamic, how does that change in terms of from a greenhouse, if we're walking now into a indoor vertical where we are sole source, right, from an LED standpoint or from a lighting standpoint, how is that lighting challenge different moving from greenhouse into an indoor vertical?
Yeah, so in greenhouse, we talk about different color or spectrum from the hand slide. If the sunlight have all the wavelength, so it can be from UV, blue, green, red, far red, and also infrared. Now going to an indoor application, then we're missing some elements. But is all element needed? It's really plant specific. We talk about some photoreceptor. Some plants are more sensitive to some photoreceptor on their growth or their structure. So again, we need to have more control on the light in an indoor application than it is on greenhouse because we count on sun. There's a plus and minus there. The minus is that, we need to control more, but the plus is that we have a better quality or uniformity of growth, because you're controlling all the elements, then you're controlling your growth, you're controlling the structure of your plant.
And so, talk to us about, I guess, indoor vertical, about, I guess, the geometry or the physics of it. I mean, it's different. We're talking about in a greenhouse, a large head space, so we got a lot of room to work with. If we're thinking about an indoor vertical, typically, head space in terms of the space from the crop to the light fixture are typically smaller. We're talking about more compact growing areas. So what are the challenges here? How would a lighting fixture design or a light plan be different when we're talking about indoor vertical versus a greenhouse?
Yeah, so again, greenhouse, you try to not hide the sunlight. So you try to be as small and light as possible above the crop. Where in indoor application, the most important is bringing the light over the whole surface where it's really shallow. You don't want, usually in indoor vertical farm, there is multiple level, and from an operation standpoint, going from, let's say, a 4 to 8 level of growing, then it's a big win for growers. Now, you want to be shallow from optimizing the space, but if you're too shallow, then the luminaire or the lighting, it's more challenged to distribute the light uniformity on the space. So that means lower power, more spread over the surface is usually better. But the bigger the fixture, the more costly. So there's trade-off depending on your infrastructure. So there's a trade-off on the size and the geometry of your luminaire for indoor.
And if we're talking about indoor versus greenhouse, we're talking about spectrums. Your spectrum, recipe will be different, or your spectrum strategy will be different. In a greenhouse, you do rely on the sun to provide you some of that spectrum, and now you're trying to level out the spectrum in your greenhouse. You're trying to level out the light level in your greenhouse. Same thing, I guess, in indoor vertical. You're trying to create a certain type of spectrum. Can you talk to us a little bit more, Dan, about when we're looking at indoor, how your spectrum selection or your strategy is slightly different from a greenhouse?
Yeah, so from a spectrum standpoint, try to be, from a luminaire design, try to be as efficient as possible and the lowest cost possible. We all heard that purple light, just blue and red light is the most efficient on young plant. And in greenhouse, there's already all the spectrums, and let's say there's already blue, red, green, far red. Now, if you just want to add more to your greenhouse, maybe just purple is good. Now, in indoor application, it depends on plants, but in indoor application, you may need some green light to get more penetration into the canopy and have a different signal or generate a different signal to the plant. Again, if you don't want to have a plant that stretch too fast, too long, and too thin, if you're really shallow, you may want to have more compact. It really depends on your sales strategy. But then there is a challenge. If you want to bring green light from a technology standpoint on LED, the green light is less efficient to generate than it is for red. And this is what we can see on the screen right now. This is kind of a typical efficacy of an LED at the nominal current. And red is the most efficient to generate photon. But then far red, and blue, it's kind of decent in efficacy. But then green, using phosphor conversion, or most of the time people are using white LED, it's less efficient. So really depend on what you need on your plant. Then you can play with these technology to generate light to your plant and have an efficient system. Also, efficiency or efficacy in this case, but the efficiency of your luminaire will affect, also heat structure in your indoor farm. If you're let's say in a greenhouse and your luminaire is not efficient, it's just the fact that you're spending more electricity to light up your surface. Because if the luminaire is generating heat, it's usually close to ceiling, and you can open the roof most of the time to get rid of the heat. Where in an indoor farm, there is so much energy need that a lower efficacy system means lower efficiency of converting photon, and also means generating more heat into your indoor application. You may have an impact on AC, on air conditioning on your indoor farm. Also, there is other. Yes, go ahead, Bruno.
Well, I just wanted to just maybe bridge the gap here. When we're talking about spectrum, we're talking about discrete colors, right? For us, just for the audience, when we're talking about a spectrum, we're talking about combination of these colors. We often hear either a purple, a pink, a white light. Can you, in terms of looking at different wavelengths or bands of wavelengths, in this case green, blue, and red, just walk us through when we're talking about a purple light, a pink light, a white light, just how those trade-offs come in terms of efficiency of the fixture.
Yeah. So again, looking at the efficiency on the screen right now, purple is about using blue and red. So you're using kind of the highest efficacy of the red and just a small amount of blue to control the structure. So it's really efficient. Now, when you try to be broader spectrum or you try to bring light that is outside of the bar, then you're moving from higher efficacy LED to lower efficacy LED. And then this is where there's an impact. The more white or the more green you put into your luminaire, the lower the efficacy usually, unless you're underdriving the LED, so using more LED but less stress, or you have a really good thermal system on your system that may bring the efficacy higher, or you have a luminaire that is just have better efficiency on extracting light from the LED to the plant. It can be, let's say, by just having more clear plastic, or have lens that bring the light at the right place on the plant. So there's no oversaturation of light in some plant and undersaturation on other plants. And the last portion is the driver and the electrical strategy. So if your driver is super efficient, or you're using a network that don't have any loss in it, then we're more efficient. Then profitability is higher.
So I guess we're talking about fixture efficiencies. There's a lot of different, a lot of different cogs, a lot of different gears that we need to work with. Spectrum being one of them, power supply being another, optical systems being another. It's a very complex system, and that's what we try to do. That's what we try to pull in all of these elements, and we try to get to the most efficient product, the most cost-effective product. But I want to pull in Hans here, because one thing is really important is that although we want to go with the most efficient light, and we want to talk about operating costs for the grower, the most efficient means spending less money for those photons. But I assume in some cases you want something that is maybe not as efficient, but you're really targeting a very specific spectrum to elicit a behavior from the plant. So can you talk to us where sometimes, maybe red, 100% red in terms of efficiency may not be the right thing for the plant, and you want to start trading off a little bit of those efficiency dollars to elicit more response from a plant. So can you talk a little bit about that?
Sure. Looking at the graph and looking at how efficient red light is. Mm-hmm. So if a single plant was just a chlorophyll molecule, mm-hmm. and red excites that chlorophyll molecule like none other, that would be ideal just to use red, 100% red. But plants are much more complicated. So they need to take cues from blues or greens and UVs and far reds to restore normal function. So if you were to just grow a plant under 100% red light, you, theoretically have very high photosynthetic rates, but you'd be missing and lacking cues from the plant, for example, properly orientating its leaf, which is perceived by blue light. And that way it will angle itself so that it can capture as much light as possible, mm-hmm. And regulate cell growth so that you will basically not have a runaway train with red light, because cells can just keep multiplying with red light. And blue will kind of tone that down and say, hey, let's not go into a hyper state here. We need to make sure we're capturing light economically and efficiently, for example.
Right. And so what's really important here is that when we're working with growers is, how do we approach this problem in terms mm-hmm. of selecting the right spectrum? What are mm-hmm. How do we approach this problem? Talking with growers.
It's tough to generalize for each given species or mm-hmm. type of plant or cultivar, mm-hmm. or even the strategy of the grower. Perhaps, they will have a certain result that they want, mm-hmm. whether it's ultra red lettuce, or maybe they have flowering that they want to keep in mind, and flowering status. So that's different examples. It's very difficult to just give a one answer solves all type of thing. Mm-hmm. So you really have to know your specialist, right? And knowing the information from your light supplier, like here at GE Current, a Daintree mm-hmm. company, and Hort Americas, mm-hmm. which are agencies and companies that are accumulating this information and trying to figure out which light recipe is the best for each given crop.
And I guess then, like, if we talk about talking about recipes for a plant, I mean, we're talking about a plant, and part of the discussion, it feels like that a recipe or a spectrum for a plant is fixed. But then there's ways of creating a light plan, let's say in an indoor vertical, that allows you to hit different spectrums at different types or different growth stages of your production. So can you talk to us a little bit? And then how do we, in an indoor vertical, how are we optimizing a light recipe? And how are we activating, I guess, these photoreceptors with our different spectrums? So can you just like visually or talk us through how a light plan looks in a indoor vertical?
Yeah, so there's three strategy for changing spectrum over on crop in an indoor application. One is about moving the plant from a section to another, and each section has a different spectrum. Second option is moving the luminaire. So you may be able to move the luminaire and have the crop in the same location. And the third one is about having a fixture that can change the spectrum and have stay in place and have the crop stay in place. There is different challenge. We all know that moving luminaire is not something that is usually user-friendly because of all the electricity and the damage you can do to the luminaire. Then there is the two other option of moving the plant if it's feasible, because it may not be suitable for all growing system and all crop. But if you're moving the plant under different lighting, that's kind of a cheap, easy way of changing the spectrum and having a different effect on your plant. As Hans said, you may want to grow your plant and then activate, the color at the end of the growth, I would say, for red colored or intense red color lettuce head. Now, the last one is tunable, or what we call tunable, changing the spectrum from a luminaire. That's really good because you can control, linearly. It's easier to tweak the spectrum. But the big challenge is if you're able to change a color, that means you have over the quantity of LED for the luminaire, and also the complexity of controlling the current in your system. But again, it really depends on your growth structure, and there is kind of really two way of controlling light on your crop.
All right, so that's really interesting. Again, this talks about I guess how dynamic and how I guess to a certain level how complicated choosing the right spectrum or right lighting recipe, and that's where having that expertise really comes from reading a lot, working with a lot of different customers, and that's what, in essence, is what we try to do, is try to understand from a research standpoint, and that's what Hans is doing, is looking at understanding what the universities are doing out there, what kind of research, what kind of white papers, and how we're pushing the boundaries of light when it relates to plants, or I guess in most cases it's just discovering new applications of light and new ways that those photoreceptors are accepting light, potentially finding photoreceptors that we didn't feel of importance or know how to activate. Is that a fair?
It's with the mass application now of LEDs and all the different diodes and colors, ranging from the visible and now from the non-visible, the far reds and the UVs. There's still a lot of open questions out there, and a lot of publications, a lot of research, a lot of experimentation, whether it be at the university or within your vertical farm.
Okay. And so I think that is, to think or expect that to come from a grower, to know and understand and be on top of all this research and understand what are the different, the latest lighting recipes or strategies. I think that's difficult to expect that from growers, and that's where we come in. That's where we definitely come in and help and support. And we also work, closely with Horde Americas, where Horde Americas is trying to pull in a lot of that application and going out to seeing growers and understanding how we could just move forward on this industry, working with growers, working with light recipes, and not only thinking about light as the end-all be-all, but understanding that growers' challenges are more than the light. We're talking about irrigation strategies. We're talking about airflow management. We're talking about a very dynamic equation. And our goal is simply just to be there as a tool and there to be as a partner and there to help growers get to the outcome that they're trying to achieve. And so with that, there's a couple of questions that are coming up here, and then I just, I'll go ahead and ask one. One that we touched on just briefly, and it came in and out, is the notion of photo period. So when to light, when not to light, or night interruption. And so Hans, can you just give just briefly, when we're talking about photo period,
sure,
just a general definition or why that's important for growers to understand.
So again, depending on your crop, different photo periods can give you very wide differing results and not always beneficial. So you really want to hone in and customize that photo period for certain crops to make sure it's responding the way that you want it to. And not even responding today, but will it respond or flower or stay vegetative when you want it to in a week or 2 weeks' time or 3 weeks' time. That's one example. Mm-hmm. Now the other balance is, okay, we didn't really talk much about DLI or the daylight integral. Mm-hmm. And do you want to concentrate your light into a short photo period, or do you want to elongate that photo period but still deliver that's the same amount of photons, but just over a longer period of time? And you can get differing responses by either ramping up or decreasing your photo period.
Okay. So here, another question that that came in here is maybe different, not necessarily tied to light per se, but talking about the notion of photoreceptors. Is light the only thing that activates a photoreceptor or elicits a response, or is this also tied to other factors?
It's probably not described for all photoreceptors at this point, but of course, plants being dependent on the local environment for their temperature, so it's now the status of the whole plant, not necessarily the status of the temperature of the photoreceptor. But there are some papers that describe how phytochrome B, which I mentioned earlier, is a photoreceptor. It actually also acts as a thermosensor. So it can sense temperature, and it can, depending on if it's a cold or warm day, create accelerated results for phytochrome activation.
Okay.
One example.
And so, there's another question that kind of builds off of this, but one thing that, is when we're talking about temperature, when we're talking about how those photoreceptors, activate that temperature, I mean, there's other ways of tricking the plant into temperature, mm-hmm. I guess. So what are the notions about, infrared, something that's outside of that PAR range, mm-hmm. that's activating? So we're talking visible and outside.
So we discussed mostly the visible spectrum. Since its importance in driving photosynthesis. But then there's also the far-red effect on promoting photosynthesis within the plant and also activating the phytochrome. Now, if you want to go even further out from far-red, then you're going into infrared territory, mm-hmm. which is also not visible. Then you're talking more about temperature impacts on the plant as opposed to visible radiation effects or photoreceptor effects, because there aren't any photoreceptors that I know of that are working in the infrared zone of radiation.
Okay, all right. And I guess with that, we're coming close to the end here. I just want to, I want to thank Hans. I want to thank Eden for taking the time to be with us today. Really what I see this is that, and we spoke to this just actually before we actually started the webinar, is that what I find so fascinating about this space is that it is so broad, and there's so much information to go get. There's so much more information to go discover that you could go deep in a lot of different places. And how we could relate that back to our customers and our growers is, given that there's so much information, where do they go get all these answers? How do they really figure out what's best for them from a lighting standpoint, from an irrigation standpoint, from an airflow management? How are they actually getting those answers? And so that's where, GE Current, a Daintree company, that's what we're trying to get at, is trying to be here as a partner for growers. We're trying to be here as a place where they could get answers to their questions. And we partner very closely with Hort Americas, which also brings on a ton of that application knowledge and really helps to move to answer those questions for growers and get growers to the next level where they want to be. So if there's any questions, anything that you're looking for in terms of our product, you could always go to our product page and see what the latest product releases are. We are releasing our 1,000-watt equivalent product that's going to be released this growing season. So very excited about that. And if you have any questions, feel free to reach out directly to us or feel free to reach out to Hort Americas, our partner. And I would like just to close on that and thank everyone for joining. And I hope this was an insightful session, and looking forward to the next one. So thank you.